Next category of assays is biological assays. Earlier we have studied the methods of quantitation of fermentation products which were classified under physicochemical assays. Now this is about biological assays, measurement or quantitation of fermentation products using biological approach. So biological assays include the quantitation of fermentation products that inhibit or stimulate the growth of a sensitive test organism. That organism is called as indicator organism. So the compounds which has a effect which has an effect either growth inhibitory or growth stimulatory. Such type of substances can be only measured or quantitated by using biological assays. These assays also include the use of enzymes for the measurement of fermentation products. These assays are called as enzymatic assays. Now let us see how exactly microorganisms are used to measure the quantity of fermentation product given in the given preparation. So biological assays are of two types, several types. First is diffusion assay. So diffusion assays, as the name indicates, diffusion. So in these types of assays, the compound is allowed to diffuse in the medium, and therefore the medium has to be in the form of solid. So these assays are carried out on solid medium, usually an agar medium, which is suitable for the growth of indicator organism or the test organism. So the example of diffusion assay will take into consideration an antibiotic assay. So antibiotics are the substances which inhibit the growth of sensitive organism. So quantity of these antibiotics in the given preparation, it can be measured by using diffusion assay. So in antibiotic assay, the test organism, it is present on the surface of solid medium. That is the medium that supports the growth of test organism. So you can see in this figure that this is the petri dish containing growth medium and on which or in which the test organism is already inoculated. This organism is should be sensitive to that particular antibiotic. So that should be a standard culture. Now there are various forms of these diffusion assays. So the material that we can use in this are either paper disc, sterile filter paper disc, then wells or cups containing antibiotics. So there can be use of paper discs, or the wells can be bored into solidified medium, and these wells are then sealed by the addition of a drop of molten agar. Then there may be use of cups, cylinders which are made up with stainless steel, which are embedded in the agar medium. So all these materials, paper disc, wells, cups, cylinders, are responsible for the holding of a known quantity of known concentration of antibiotic solution. So these are the materials and methods for antibiotic assay. Then the next step will be we must have a standard reference compound, as we have learned in case of spectrophotometric assays or earlier colorimetric assays, that there is a need of standard reference compound. So that standard reference compound has to be there, so that with the help of that we can prepare a standard graph. So the protocol is same. With the help of the standard reference compound, first step is to prepare dilutions of that particular antibiotic. So these are the tubes containing the antibiotic, and this antibiotic solution, it is diluted so as to obtain various dilutions which are marked here, like 10 micrograms per ml, 20, 30, 40, 60, and so on and so forth. So this standard antibiotic solution is diluted so as to get these different concentrations. Then with the help of this disc, we immerse that disc into the first dilution, that is 10 micrograms per ml, and after that, that disc is then placed on this agar surface. If it is well, then the first dilution, approximately 10 to 20 microliters of this, it is then added to this vial or cup or cylinder. Likewise, it is repeated for 20, 30, 40, 50 and 60. So discs are immersed in the respective solutions, and then they are placed onto this solidified agar. So this is how we can call these modifications as disc diffusion assay or well diffusion assay or cylinder or cup diffusion assay. So after placing all these antibiotic containing discs of different concentrations on solidified medium, the next step is incubation. This plate is then incubated at desirable temperature, the temperature and the conditions which are favorable for the growth of test organism, that is antibiotic sensitive organism. We'll get the results like this. So you can see that the disc containing 10 micrograms per ml will show a zone of inhibition. You can see wherever the antibiotic is absent, there you'll find the presence of growth of test organism, and the zone around the disc where antibiotic is diffused, there will be absence of any growth of the organism. It is called as inhibition zone. So these inhibition zones are produced in the circle because antibiotic solution or antibiotic, it diffuses radially, and therefore will get this kind of circular zone. Now we have two parameters. First is we know the concentration of standard antibiotic, and second parameter, now we have the data regarding the inhibition zone. Now how to record this inhibition zone? Inhibition zone is recorded in the form of its diameter. So now you have inhibition zone diameter and corresponding antibiotic concentration. Now you have fermentation broth containing antibiotic, so that is the sample. So this earlier A part, it will be used as a standard set of conditions, and now you have sample. Now sample has to be processed in the similar manner. So a disc is immersed in this sample, and then it will be placed on the similar solidified medium containing test organism. Again that plate will be incubated and it will generate some inhibition zone diameter. like this. Then you have to measure the inhibition zone diameter like this in centimeters or in mm. So it will be called as the unknown diameter. Now you want to convert this diameter reading into concentration. So it has to be compared with the set of standard. For this, we'll prepare a standard graph because now you have two parameters, antibiotic concentration and inhibition zone diameter. So inhibition zone diameter is on x-axis and log of antibiotic concentration, that is in micrograms per ml, it is on y-axis. So you know both the values, so all these values are plotted onto the graph so that it will follow a linear curve. That means as the concentration increases, the inhibition zone diameter increases. Then we have to draw a line that will pass through these points. So this will be an average line that will try to accommodate the two most divergent points. So this adjacent line has to be drawn so that all the points should be maximally covered. This is called as best fit line. So once you get this best fit line, now in earlier slide, you know the unknown diameter of the fermentation broth antibiotic. So that inhibition zone diameter, it will be plotted onto this graph on the linear part so that you can convert this inhibition zone diameter into antibiotic concentration. So this is how this standard graph will be useful for conversion of inhibition zone diameter readings into the concentration. This is how we determine the concentration of antibiotic present in the given preparation. Likewise, the growth stimulatory substances, for example, vitamins. So vitamins which stimulate the growth of indicator organism. So these vitamins concentration, it can be determined by using this diffusion assay. Only the difference will be that antibiotics are growth inhibitory substances, and so which are likely to generate inhibition zones against the producer organism, against the indicator organism. Whereas in case of vitamin assay, the indicator organism is chosen such that it will require that particular vitamin, and accordingly it will exhibit a zone of exhibition. So that will be the difference between antibiotics. So in antibiotic assay we measure zone of inhibition, and in vitamin assay we measure zone of exhibition, means the presence of growth. Protocol is same. You must have a standard reference compound, that is vitamin. That vitamin is then diluted, say for example from 10 to 60 micrograms per ml. Then you have unknown, that is fermentation broth, in which the vitamin is present. So that is unknown. So following the similar protocol what we have studied in case of antibiotic assay, same protocol, that is diffusion assay, we can use. Say for example disc diffusion assay. So the discs are dipped in corresponding vitamin solutions, and then they are placed onto the medium containing an indicator organism. Now in this case the indicator organism has to be an auxotropic mutant. Auxotropic mutant means this will be the mutant that should require this particular vitamin for the growth. So in case of this type of assay, once the plate is incubated at desirable temperature, what we are going to find is presence of zone of exhibition. So wherever the vitamin is diffused, there only you will find the presence of growth of this auxotropic mutant. Rest of the part will not show any growth of this particular mutant. So zone of diameter of zone of exhibition, here in this case we have to measure. In this case the graph will be like this, that zone diameter, that is exhibition zone diameter, versus concentration of this vitamin. So as the concentration increases, the zone of exhibition increases. Then we have to prepare a best fit line. Once the best fit line is generated, then you can measure the zone of exhibition produced by unknown sample and convert that reading zone of exhibition diameter into concentration. This is how we can determine concentration of unknown vitamin in the given preparation.